And we are going to actually, whoops, I'm going to share my screen so we can view the lab together. If you have your own copy of the lab, you might want to have it open to figure out what we're doing. All right, so this lab is called Relationships and Biodiversity. And basically what we're going to do is we're going to be investigating the difference between structural and molecular evidence. So there's this plant, this is made up, by the way, this isn't real, but it could be real, right? There's this plant called Botana curis, which very clever, oh wait, you guys can't hear me. Uh-oh.
Are you muted?
Can other people hear me? I'm not muted, and I have my volume all the way up. Okay, you, Peyton and Samantha, try like muting and then unmuting, see if that helps you. You might leave the session and come back in, too. This is also being recorded. I know they can't, oh yeah, they can't hear me. Okay, sorry guys. Try to leave and re-enter. Okay. Wow, that's weird. Some people can't hear me, but some people can. Okay. So this is also being recorded. So if you can't hear part of it or something like that, you can always go back. Okay. So Botana curis is this imaginary made-up plant, right, that cures cancer. So what we want to do is, because we're using it a lot, obviously, people are over cultivating it, right? They're taking it out of its natural habitat and it's not able to reproduce enough to actually stay caught up with the need for it. So we're going to be looking at 3 different plant species, X, Y, and Z, and we're going to try to determine which one is most similar to Botana curis and therefore has the most potential to also be a cancer-curing plant, right? So hopefully you guys already looked through this lab and tried to answer the pre-lab questions on your own, but just in case you didn't, we're going to talk about them. If you're in the Zoom session and you want to type in the chat, you can. But what are you guys feeling in terms of which evidence is going to be the most helpful? Structural evidence or molecular, right? In structural, we're talking about things like what the plant looks like, the color of the plant, the stem. Molecular, we're talking about stuff like DNA, proteins, etc. Okay. Who's entering unmuted here? There we go. All right. Hold on 1 second. Okay. So, what do you guys think? Structural or molecular? Which one's probably going to be better evidence? Molecular, right? Hopefully a few people are also thinking that in their minds, right? Just because a plant looks like another plant doesn't necessarily mean that they're that similar, right? Think about it. If you went out into nature and you're looking at plants, you're like, oh, look at these plants, right? You're not like, whoa, this one looks so much different than this one. Unless you're like a plant nerd and you know about plants, right? Which is good for you, but they look like plants, right? Whatever. Molecular is going to tell us more about the DNA, right? And the proteins, which is going to be able to tell us more about if that plant could actually cure cancer. So you were also supposed to review some vocabulary, right? A lot of this was from last semester: enzyme, DNA, RNA, protein, amino acid. The reason why is we're going to talk about those things, right? It's going to be a review. I know those are hard molecular terms, right? So don't worry, I'm here to guide you through and do the lab with you. So I love this lab. It's one of my favorite ones. Okay. And then major structures found in a plant stem, right? Just something for you to review. Really, you don't need to know specifically like every part of the plant stem, but you want to realize, right, plant stems do look different, which might come as a shock to people, but they're arranged differently, right? Depending on the species and depending on how big the plant is, and also how complicated the plant is. So what we're gonna do for part 1 is, if you're here in person, we have these nice handouts that tell us about each plant species, showing us a picture of the plant, showing us a picture of the plant stem, and then telling you which species it is, right? We also have seeds from the plants. If you are at home, you are going to scroll to the bottom of my original biology lab document, and you will see lots of helpful things. You'll see pictures of the plants. I didn't put them in order, sorry. But I labeled them species Y, species Z, Botanicurus, species X. And then you'll see the stems, right? And then you'll see the seeds. High quality pictures taken right there. So what we're gonna do is we're just gonna spend a few minutes here in your data table. You're actually gonna draw what you see. And I know you're thinking, well, why can't I just copy and paste the picture in? Right, I want you to draw, because if you're drawing it, you're actually gonna spend time looking and thinking about what you're looking at, right? If you're copying and pasting the picture, you're like, oh, it's a plant, oh, it's a stem. No, I want you to think about what it looks like. Look at the veins, look at the way the leaf looks, right? You're looking for similarities here between these plant species. So if you're at home, I encourage you to draw, even if you're drawing in Google Drawing, which would actually be really hard to do this, but you could try. Or just sketch it on some paper, thinking about the differences. You're possibly gonna have to open this document on your own, because there's not an easy way really for me to show all of them at the same time. Right, but we're gonna spend, I don't know, probably about 5 minutes right now looking at the different plant samples and sketching in part 1 of the data table, okay? So you guys can, yeah, you guys can grab at your leisure here. And I have some colored stuff here too if you want to make it look pretty. Okay, let's see. Okay, audio is working. Samantha release. What's that beeping?
That has been irritating me for the last 10 minutes.
It's weird.
Going on for a long time.
Okay, the pen's fine. And while you're drawing the plants, fun times, and looking at all the differences between them, I want you to be thinking which plant species looks the most like Botanicurus. That's gonna drive me nuts.
You're like, let's fix this.
Yeah. Well, it's either a really hot room or beeping.
Oh my.
Like backing up.
Backing up for this long?
It's like sitting there, like slowly moving to go forward, and then like slowing moving. Maybe it's like broken or something.
It's like broken or something. Yeah.
Oh my. UPS.
I just realized that I wrote that super crooked.
Okay.
That is like crooked.
Everyone knows that problem.
That's my
Writing's hard.
I'm about to look along doing this.
Yes.
I'm gonna try to have you position the computer so that I can see that. I don't even know if that's gonna be possible. It might be backwards.
I think it's too small. Trying to see that. I think it is it reading it backwards? Yeah, I think it is. I think it's backwards.
Yeah, it is. Okay.
I could take a picture of it.
Then maybe I'll just have you write in the document while I talk.
Okay. Do I need editing rights?
You can you can do it from here. And then you can tell me if people are asking questions too.
Do you want to just write everything on there in the document?
No. So
Oh, it's already in.
yeah, so when I'm writing, you're just gonna write what I write.
Okay.
Yeah.
Oh, let's check.
Okay. But we're gonna do that stuff first, actually.
Oh, okay.
All right, so sorry guys, if you're tuning in from home, people are sketching plants right now. We're just trying to figure out what the best way to show this stuff is so that you can see it. Hey, it's okay. It's not an art class. Like I said, the art of drawing in science is not so much about producing beautiful art, but thinking about what you're drawing and what it looks like.
My skills.
So it's
Like,
all right. So I'll take this time to remind everyone. May 9th, we are dissecting.
I'm ready for that.
And I ordered a frog, a rat, a pig, and a starfish.
Whoa. I'm actually surprised because a lot of people just do frogs.
Oh, no, we're having a variety.
That's fun.
So make sure that you have that on your calendar. Make every effort to come in, right? I will record it, but obviously it's not going to be the same. And it is a cool experience. And if you're scared of it, it's kind of weird at first, but I'll cut it open for you if you need me to. And then after that, it's really cool. And the starfish is cool because it's 90% reproductive organs, which is weird.
Which is weird.
Yeah.
You're like, okay.
I know. You're like, all right, starfish, like clearly you have one purpose here, reproducing. That's it. It's true.
Good job, starfish.
Well, starfish are animals. So they're like simple animals. Yeah. Not as simple as sponges, but sponges aren't very fun to dissect. You can do that. You can take a loofah and cut it in half. That's dissecting it. Yeah. Oh, beautiful. Okay. So people are starting to finish up drawing here. So looking at the plants, right? We have Y, Z, X, and Botanicurus. And yes, I just took plant pictures from Google Images. This was a lot of work for me, guys, to create this. I'm telling you, it takes a lot of planning. And then we have different stem samples, right? Some really simple, like species Z has a very simple stem. Species Y's stem is very intense, right? And then species X and Botanicurus look oddly similar. And then we have the seeds, right? X is next to Botanicurus in this picture for a reason. And Z and Y. Y's seeds definitely look the most different, I would say. So those of you who've been sketching and thinking about this so far, which plant is looking like it's the most similar to Botanicurus?
X.
X. Yeah, I would agree. So there's actually a place for you in the document, which this document is a lot of pages. But in part 1, where it says structures of plants, question 2 says, which plant, after looking at the structural features, is most similar to Botanicurus, right? You may say plant X if you think something else. Where can you download the document? It was in an email, but here, I'll send you guys the link again. Okay. Liv, yes, if you make a copy of the document, you can put your drawing into your own document because you're going to be turning this in. Okay. So most people are feeling species X, right? Especially, I would say, from the stem. The stem looks very similar on Botanicurus and species X, right? The seeds look very similar. So those are good indicators that possibly they are related. The next part of this, we're going to be testing for enzyme M. So quick little review. What is an enzyme? It's a protein.
I thought it was that. I just thought I was wrong.
And it helps speed up reactions, right? So those of you who've been here the whole year might remember when we did the chicken liver enzyme lab. We cut up pieces of chicken liver and we poured hydrogen peroxide on it and we put it at different temperatures to see the reaction, right? So enzymes need specific conditions. They need a specific temperature, they need a specific salt level and a specific pH level. And if any of those things are off, they don't work. Right? So you can think about them like, they're almost like fragile, like an ice cube, right? If they're in the freezer, they look like an ice cube. As soon as you take them out of that, they're melting. And they do, they change shape and get distorted and then they don't work. So enzymes are super useful, right? In this case, enzyme M is part of what helps treat cancer from Botanicurus. So we're gonna test samples from each plant and see which other plants have enzyme M, right? Because if they have enzyme M, there's a good chance that they will be able to also cure cancer. So the way that we're going to be testing this, and go ahead and keep drawing as long as you need to, is we have some cups of plant samples. Let's see if I can show this without pouring it on my computer, which I've almost done before. We have different plant samples here taken from each plant, and we are going to be gathering around to test these plant samples. We have these little spot trays. They're called spot trays because they have spots on them. Imagine that. We have this white powder. That is our indicator powder. That is going to tell us if enzyme M is present. So how would we know maybe that a reaction is happening? Like what would we see if something is reacting?
Changing color.
Changing color, possibly.
Consistency, maybe.
Yeah, maybe like getting thicker or thinner, right? Bubbling is a good indicator. A weird smell. There won't be any weird smells, don't worry. But those are all good indicators. So that a reaction is happening. So here is our indicator to determine if enzyme M is present. We have our cups of plant samples. And this was, I guess, nothing. This, okay. So what we're gonna do is, I'm gonna have one person take just a little scoop of the powder and place it in here. I'll do the first one. Let's test Botanicurus first. In our data table, by the way, we're going to be writing present or absent. That's pretty easy. So we're going to just squirt in some Botanicurus sample. Actually, let's see if we can get this in the view of the camera. Okay, are we ready? What do we think? Enzyme M is present or absent? Present, right? It reacted. There was a bunch of bubbling. It made a hissing sound. It was like, all right. Who would like to scoop some powder for the next one? Or I can do it if you don't want to. Okay. It's not going to hurt you. It's a harmless powdery substance. Just go ahead and place it in one of those guys, and we'll do X next. So notice X is a different color. Go ahead and squirt some X in there. Let's see what happens. A little bubbling, right? Not as much. It was like bzz, kind of bubbling. Okay, next we'll do species Y. Species Y, I think, is the prettiest color. Do we see any reaction? Nothing, right? Species Y, it did nothing. Okay. And lastly, species Z. You want to do Z? Good job, species Y. Well, so what is that telling us? That's telling us species Y does not have the enzyme. So probably not the closest related. We'll see. There are many tests yet to come. So could be. That's enough. You don't need too much. Yeah. What do we think? Present or absent? Present. So in our data table, Sydney, I will talk about that at the end. Okay, in our data table, I'm going to just write on the annotations here because I don't want to fill in the lab document. We're going to be putting for enzyme M, we're going to put that Botanicurus enzyme M was present. For X, it was kinda present. Scientific language, I know. For species Y, it was absent. And for species Z, it was present. So the reason too that we're doing so many different tests is because just like when you test for anything, right? Even if you're thinking like criminal justice, we want to be sure which plant is going to be correct before we start wasting resources to harvest it and make it into a cancer drug, because that costs a lot of money. All right, up next, we're going to do paper chromatography, which is really fun. So this is an actual chemistry technique. This is my favorite thing. We're going to be taking these round filter papers, and we're going to place drops of each sample of plant on the filter paper. We're going to dip it in water, and then we're actually going to get a cool pattern, which they will see later. We're going to get a cool pattern of the pigments of the plant, right? And it's just another way to separate out a sample to sort of tell us if it's made up of the same stuff. So I want you guys to draw a line on the bottom of your filter paper and label it BC XYZ. You do want to do this in pencil. So if you don't have a pencil, wait to borrow a pencil. Those of you who are at home, there is an example of this chromatography at the very bottom of the document that you will see in a moment. Okay, then you're going to take just a few drops of each sample and just place them right above where you labeled.
Okay. Thank you. Okay.
Yeah, make sure you read which one it is too. Otherwise,
the wrong one.
Otherwise, it's going to be all Botanicurus, and you're going to be like, what? I don't know. Okay. And then Elizabeth will help you guys put this in the water.
Many drops, like
It's pretty self-explanatory. But you can also do this with markers, which looks cool if you ever want to do like a fun art project.
I almost tried to put the same one in.
And here, you guys can come over here and start grabbing them too. You don't have to do a one at a time here.
Why is he? All right, take one for half a second. Thank you. There you go, and then the water.
Dip it.
It's like,
Thank you. Yeah.
then we hang up to dry. B, C, and D are hooking up. That's not what I meant to say, but that's the thing that came to mind.
That was perfect.
Yeah. They'll hook up later, I guess.
Whoa.
I know you're.
Please no.
Why was it? Still a little over here.
And so at the end of this, you end up getting something that looks like this.
None of the Z's are turned out quite like that one.
Yeah, well, that's the interesting thing about chromatography as well, is that it can be different every time. Oh yeah. Those Z's aren't looking as beautiful, but that's okay.
That's beautiful. Good. Do we hang it up too?
You can if you want to let it dry, and then you're welcome. You're welcome to take it home. Keep it as a souvenir of biology.
I did sign.
Yeah, you're like,
Congratulations.
I did chromatography today. Well, then you can explain it to them. It's the fun part about science. So again, those of you who are tuning in from home, if you scroll down to the bottom of the document, I included a picture of my chromatography. And guess what you're gonna do with it? You're gonna draw it.
Wow.
Although if you're here in person, you don't have to draw it. You can just staple it to your lab. That's fine, or include a picture of it because you're doing it in person. So looking at those samples, stop playing with those.
I'm trying to get it back into the thing.
Enzyme M's being played with over here. So we're going to be looking at this as one of our tests to see which plant is closest. Right? Our next test that we're going to do, which by the way, we're kind of mimicking real science here in the sense that real biologists are going to be doing tests on a bunch of different stuff, right? To find out which plant is similar, we wouldn't just do one thing, which is why we're doing like 20 things. All right. So what we're going to do next is the hardest part, I would say. And that is the amino acid sequences, translating amino acids to proteins, which you're lucky your body does that for you. But we're going to learn how to do it and how the body does it. So everybody's going to need to open one of these universal genetic code charts. And people who are here will look at it together. But we're going to need that for the last step. We're also going to, I'm going to be doing this on a board, which will be backwards for people at home. So Elizabeth is going to be typing what I write on the board, but you'll be able to hear me. So it's going to be kind of strange, but it's going to work, I think. So if you guys want to write this down as well, just on a piece of paper, or write down how to do it so that you could do it later, that's fine. What happened to the pink? Okay. So first thing that we want to know / remember is that we have these letters that code for DNA and for RNA. Right? And this is probably review from last semester, or maybe you never learned that and now you're learning it for the first time. which is possible too. So with DNA, we have A, T, C, and G, right? RNA is the same, except that instead of a T, we have a U, which stands for uracil, right? RNA doesn't use T's. It's just a different coding. So what happens when we do protein synthesis, which again is something we learned about last semester, is the body takes your DNA sequence and it gets converted into RNA, and then it gets converted into a protein, right? And most of us, if you did the assignment for this, it was like, describe how it happens. So some people probably have never done this before. It is a little confusing. Bear with me. It's fun. You get to use a chart. Okay, so all we do, literally, is we copy the DNA, we pair it with what RNA base it would get. A always pairs with U, which means U always pairs with A, and C pairs with G, right? That's pretty much all we have to know. So if we're doing Botanicurus for our sequence of bases in mRNA, our first thing that we're going to get here is G, U, G, C, A, C, C, U, G, A, C, U. Oh, this is going to be C, C, U, G, A, G, and G, A, G. Right? I went fast because I already know how to do this, but all I'm doing is translating DNA to RNA. They gave us the DNA. I'm just translating what that would be in RNA. Right? The body does that in like 3 seconds. It's like, it's done. You're going to do it for the other ones, right? Following the same principle that C goes with G, A goes with U. This is where it is confusing though. T goes with A. Which is a little confusing. Do the best you can with it. I just want you guys to be exposed to this concept, right? And then the next and final step is we have to figure out which amino acids this would make. So the interesting thing about amino acids is that we read 3 bases at a time, which is why on your paper they're separated into 3. So 3 bases equals 1 amino acid. So if we look on our amino acid chart, it actually tells you first base, second base, third base. And we're not talking about baseball here. We're talking about DNA to proteins. And we're just going to follow what it says. So, in fact, I probably should have printed this for people that are here. I didn't think about that. All right, our first base, for example, for our first one is G. So we find a G. Our second base is U. Okay, we're in the U. And our third base is a G. So we're going to be looking at the last one, GUG. That codes for the amino acid VAL. So on our paper, we're going to write VAL, right? Which I actually don't even know which amino acid that stands for. But you could look up which amino acid that stands for, right? Our next one is CAC. So we're going to find C, A, C, and we see that it's HIS, which stands, I believe, for like histine or something like that. We're going to print this chart out so that we can work on it over there together. Can you print it? Mm-hmm. Okay. So basically all you're going to be doing is going through, translating DNA to RNA, and then taking a group of 3 from the RNA to translate it to the protein. At the end of this, you're going to see the differences, right? So Botanicurus is going to have 1, 2, 3, 4, 5, 6, 7 amino acids.
How many copies?
Just print 3. And we're going to see how many differences species X, Y, and Z have, right? So they might have all the same amino acids, Now, what's interesting about this is when you see the chart, those of you who are here in person and those of you online who can already see it, more than one combination codes for an amino acid, right? So VAL, there's actually 4 combinations you can have that give you that, which is crazy because that means that a mutation might actually do nothing. If this gets mutated to a C, GUC, right, that might not change the amino acid, might not matter. Or it might change it and then it's completely ruined, right? So there's kind of like this weird, some amino acids have like 5 or 6 ways they can be coded for, which is pretty cool. So we're gonna work on this a little bit together. And then we're gonna take a little break though. I'm gonna talk about the gel electrophoresis so that we can end the virtual session and then just work in person here so that we don't have to like have an awkward waiting around time thing. So those of us who are tuned in from home, you guys are gonna be trying to do this on your own, right? It is hard. I will actually fill out the entire Botanicurus one so that you can use it as an example, and I'll be doing that in a moment. Otherwise, we're gonna talk about gel electrophoresis, which hopefully you watched the videos on it. It's really fun to do, but it's really hard. And the gel is like, imagine clear Jell-O. And when you're putting in the DNA, you can easily stab the gel accidentally, which ruins it. It's a very delicate process. So I did print out one gel here that we can look at. But basically, it's another way to measure similarities between species, right? All you do is you put the DNA in these top, they're called wells, and the DNA has already been broken up into pieces, right? We use another enzyme, enzymes coming back, restriction enzymes, they cut the DNA up. The DNA is gonna move through the gel, right? Why? Because it's charged. DNA is negatively charged. So if we put a positive charge at the end of the gel, the DNA wants to move towards the positive charge. Now, if you can imagine what it's like to try to walk through Jell-O, right? It's not easy. Especially you can imagine if you are taller, right? It's gonna be even harder to move through the Jell-O than if maybe a mouse is trying to move through Jell-O. It's probably gonna have an easier time. Same thing happens with DNA, right? Big pieces of DNA barely move. So these pieces that we see at the top of the gel, which I'll show you guys this again too, those pieces do not move very far because they're huge. The pieces that make it to the end are tiny, tiny, tiny pieces of DNA. So basically, this is the kind of thing that's used also for like Maury, when they do like the who's the daddy testing, right? They'll actually run a gel of DNA, and people, like offspring of parents, right? A child is gonna have all of their bands of DNA either come from the mom or the dad, which is crazy because then you can have multiple people there and you can see who the actual biological father or mother or whoever is, right? So this is used in other ways. You can also use this in crime scenes to find out who killed someone, right? If you have a sample of blood, you could get DNA from that. You can run a gel with your suspects and you can see whose DNA matches. So in this case, we're just gonna be comparing Botanicurus to X, Y, and Z and see which one it has the most in common with. Just from looking at it, you can probably tell. In your data table, I would like for you to describe the number of bands that it has in common, right? Or you can draw it, but some way for you to be able to quickly look at it and tell which plant species Botanicurus is closest to. All right, so if you're tuned in from home, you're gonna have to continue working on this by yourself because you're gonna have to do all this DNA, RNA, protein stuff. And then there's some conclusion questions. Oops, I just passed them. There are 6 questions. And number 6 is really hard because it's drawing a cladogram. Again, do the best you can. So people are also asking where you turn this in. right? If we go to the course, I will actually activate the assignment now because it wasn't activated before. But this is going to be Biology Lab 3. Let's see. 418 Biology Lab. And I'm going to put a link to this document and allow you to actually be able to turn that in. So you should be able to turn it in at the end of the day. I'm also going to upload this recording. So if you missed any of this or you want to go back and watch it again, then there you go. All right, otherwise I'm going to stop sharing my screen. I'm going to end the meeting, and please email me if you have any questions.